In this tutorial, we will look at cluster analysis in IBD. Clustering is the classification of objects into different groups, or more precisely, the partitioning of a dataset into subsets or clusters so that the data in each subset shares some common trait, often proximity according to some defined distance measure. Hierarchical clustering arranges objects in a hierarchy with a tree-like structure called a dendrogram, or tree graph that represents the similarities between objects. When clustering genes, all features or samples are often used. However, when clustering samples, the full profiles have a very high dimension, as there are typically thousands of genes represented on an array. Therefore, one might want to use only a portion of them. The other parameters that we need to select are the clustering method and similarity measures. The overall results of our analysis will vary depending on the features we choose to cluster and the clustering and similarity measure we define for this process. In each case, a different facet of the relationships in the samples will be brought to light. Please refer to pages 251 to 270 in the user manual for a further explanation of these clustering methods and similarity measures. Once we have specified our methodology, we check the box for Z-score normalization. This will standardize the data across a wide range of experiments as well as allow the comparison of microarray data independent of the original hybridization intensities. We will check the box for calculate column dendrograms to cluster across samples. Once clustering has been computed, the hierarchical cluster heat map and data expression matrix will be displayed. This diagram shows the cluster across genes and samples, and the number of clusters depends on the cutting level we set. As a default, probe set ID names are labeled on the dendrogram, but these can easily be changed to gene symbols. Additionally, we can change the grouping columns if we choose to. This data is also displayed in the filtered expression matrix and is synchronized to the above dendrogram. For further information and tutorials, please visit www.integromics.com